how do scientists think about and explore the two most fundamental questions we face? What are the origins of human life, and are we alone? Are we the only intelligent life? I see the Origins Project as designed not to answer those questions, because obviously we don't know them, but to show the public how scientists think about these issues.
Hi everyone, thank you so much for being here today. I want to start with a story of my first time looking through a telescope, which was actually almost exactly a year ago today. And the very first thing that I saw was Jupiter. In this beigeish brown sphere, you could actually see the lines of the different parts of the atmosphere, and these four little dots of light that are the Galilean moons orbiting around it. And the first thing I thought, which I'm sort of embarrassed to say, is it looks just like the pictures. Which obviously, I don't think that NASA is making up the photos that they've showed us. But I've been covering science for a while, and I've seen so many images of Jupiter. But for the first time, I was actually looking at a planet in real time. And we live in a universe rich with other worlds, and weird and compelling places. There's moons of planets in our own solar system that have oceans and potentially the conditions where life could, microbial life could live today. There's Mars, which at one point had an atmosphere and water, and we think there could have been life there, potentially, maybe. And then there's exoplanets, thousands of them. I mean, thousands that we know of and countless more that we don't. And we're detecting more every day. And on top of all of that, there's also the possibility that someone else is out there, someone like us, intelligent, seeking, exploring, and maybe sending out signals that we could potentially receive. And I actually have been thinking a lot today about something that I heard Chris German, who's a deep ocean researcher at the Woods Hole Oceanographic Institution, who is sort of working on what we can apply from our own ocean to places like Europa and Enceladus that have oceans beneath icy crusts. And he said that the search for life elsewhere is like building cathedrals in the medieval era. It was something that took centuries, and one generation would build the foundations, the next generation would build the walls, and maybe if they were lucky, their grandchildren or their great-grandchildren would get to sit in the pews. And the research that happens right now today is laying the foundations for discoveries of the future, things that maybe I won't get to report, but if I have a grandchild, they will get to report the news. Or maybe we'll find it out sooner. But I think that that's an idea that I want you to hold in your mind tonight, thinking about laying the foundation for an effort that is going to take a century, even more. And with that, I want to introduce our panelists. They're a really brilliant group of people, and I've really enjoyed getting to hear them talk today, and I think you will too. Ken Mary Wojtek from NASA's Astrobiology Institute, Ken Williford from the Jet Propulsion Laboratory, Jada Arney and Sean DeMago-Goldman from NASA Goddard, Olivier Guyon of the University of Arizona, and Jill Tarter from the SETI Institute.
So the two most exciting discoveries that have actually shaped my career was the first one in 1977, when the submersible Alvin actually came upon a hydrothermal vent system at the bottom of the ocean. This is a system far away from sunlight that supports an oasis. That was the first indication that we had that life goes well beyond what our conventional thinking about where it could inhabit places on Earth. And then the second one was in 1996, when there was an announcement from the White House that we had discovered Martian life in a meteorite. And although the Allen Hills meteorite findings didn't actually achieve that, it set up a whole new area of research and a new set of exciting tools to be developed to enhance our ability to look for life beyond Earth. So how do we look for life beyond Earth? First off, we start with the life that we know. Clearly life is all over our planet, and what can we learn about the life that's on this planet that can inform how we are going to look for it elsewhere? We know life is tough. Everywhere we look on this planet, no matter if it's in a pool that has the pH of battery acid, or inside your oven after it's been cleaned with oven cleaner at a very high pH, or down at the bottom of the ocean at high pressure, high temperature, low temperature, everywhere we look, we find life. And these organisms that live in these environments are called extremophiles. In addition, life is tenacious. So even if they aren't active at that moment, there are conditions on Earth that are even more extreme than some of these extreme environments, where there's absolutely no water, and water is necessary for life, where they will survive until the moment that they get a little bit of water. So they're tenacious. They can actually have very long survival times. Life is metabolically diverse. That means it could eat anything and breathe anything. Every time we think about a chemical bond that could be broken that organisms could use to get energy from, we find an organism that can do it. And one of the most interesting things is very recently it was discovered that organisms that were out in California and Silicon Valley were able to live on breaking the bonds of the silicon disks and chips that were actually coming from disposed computers. So they keep up with it. There's no challenge too big for a microbe. And in addition, when things get really bad on the surface, they go into rocks. So in extremely harsh conditions, like down in the dry valleys of Antarctica, you'll actually find life inside of the rocks. Now, one of the biggest challenges for looking for life elsewhere is for us to get clear on a definition of what is life. I think everybody would agree that this picture that I'm showing here shows all sorts of charismatic megafauna. We all recognize this as life. You might like whales or elephants or giraffes. But most of life on Earth looks more like this. These are diatoms from the ocean. Or like this. These are dinoflagellates that live in aquatic systems as well. Or they might look like this. This is a macroscopic version of microscopic organisms that live in one of the most diverse environments on our planet, your belly button. And many of you may remember in 1996, President Clinton from the Rose Garden made the announcement that scientists had found evidence of Martian life in this meteorite that was collected from Antarctica. And this announcement was based on 4 pieces of evidence. 2 of them I have shown here. The one well, first of all, that's the meteorite that fell in Antarctica on the top left. On the lower left is something that looks remarkably like microbial cells. There are bacteria that look like that. The problem is the size of this is about 1/100th of the size of a bacteria. And this is a problem because bacteria need to have its DNA inside and all the enzymes and all the chemistry that it needs to have to actually sustain itself. So this actually image of this structure can't possibly be a cell because it's just too small. And then on the right, there's a picture of magnetosomes. The lower one is from the rock. The upper one is from actually a magnetotactic bacteria that has that. Again, they look very similar. But the important thing that happened after this announcement is the scientific community came out and said every one of these pieces of evidence is ambiguous. We can come up with reasons why this can't be biology. We can show you examples of how I can produce this very feature in my lab, abiologically. And so even with 4 solid pieces of evidence, they thought, and really good researchers, they still had made a mistake in identifying or declaring life. And it was mainly because before that time we thought we would know it when we would see it, just like pornography. We'll recognize it when we see it, but in fact it turned out we had not done our homework and we had not studied it sufficiently. And out of that discovery was born NASA's astrobiology program. And we went at it hard to try to get more rigorous about how we looked for evidence of life. Now I want to just end my talk with talking about some of the targets. Of course, we're looking at our nearest neighbor, and Ken's going to talk to you about what we're doing next on Mars. It's very much like Earth in the sense that we thought at some point in time it was warmer and wetter and could have supported life. And then these ocean worlds that Sarah made reference to. These are the moons of Saturn, Jupiter, and there's one of Triton in there. This is their actual sizes in comparison to Earth. And the reason I put this up there is to also say that this is their size, but this is a slide of how much water those tiny moons, again, look at the volume, have as part of their composition. So there's actually, in the grand scheme of things, Earth isn't very watery. It isn't really an ocean world because it has relatively little water compared to some of these other bodies in our solar system. And then I'm going to end with another headline from work that Sarah has done, which all of you may have read, that scientists discover new evidence of plumes on Europa. Europa is one of our targets. It's a place that NASA has planned to send a flyby mission in the near future, and maybe even a lander. And this is exciting because we think these plumes are expression of the subsurface ocean, and this might be a great place to look for life. So thank you.
Great. So if anyone has a question, put up your hand and someone will come around with a microphone.
What about the Chinese and the Indians? Are they participating in some way?
The Indians are. So, they actually launched an orbiter to go to Mars to actually look for trace gases as well, and we assisted in helping with their navigation. It was a test mission called MOM. And they have plans to do other things, and we're in, what you're saying is this adventure, this enterprise, it needs to be global. And I think that the major space agencies are talking to one another about how to share resources, and more often than not, one agency will be the prime on a mission and get instruments from other agencies to fly in that. There's a mission from JAXA going to study, it's a sample return mission from the moons of Mars to understand the origin of the moons. And we're flying instruments on that. And then the other thing I think that the global science community does that's really important is we have participating scientist programs. So no matter who is flying the mission, they want to bring in all the potential intellectual interest in the data that they're accumulating and collecting through these missions, and share it with all of the scientists around the globe to be able to analyze this. And so, within what one can do across national boundaries, I think that space has been one of the places where we've recently certainly have chosen to collaborate and for the betterment of science.
We live in this incredibly exciting time where we have the luxury of a mission that is the first leg of Mars sample return. We're talking about that as a gateway to sending humans to Mars, the first human beings on another planet. We're exploring ocean worlds like Europa and Enceladus, looking for life that might be currently alive in our solar system below the ice layer on these other worlds. The fact that we are actually pursuing all of these multiple approaches at the same time is just incredible to me. Today, I want to tell you about the next Mars rover that we're planning to send. And that mission is called Mars 2020. We are planning to launch in 2020, and we're going to keep that name for the mission, so we have no excuse to miss that launch window. I just want to take the next few minutes to introduce you generally to the mission, and I hope we'll have some more time to talk about what we're doing later in the evening. So as I said, we plan to launch in July 2020, July or August. We hope it looks exactly like this, what you see here in the left panel. This is the MSL launch when the Curiosity rover took off for Mars on an Atlas V 541 rocket. We're going to use the same launch vehicle. At which point, after a successful launch, we will travel between Earth and Mars for 7 months to arrive around February 2021. And we will begin the very exciting and terrifying entry, descent, and landing process, or what we call EDL. Okay, and so if you haven't seen this, there's a great YouTube video called 7 Minutes of Terror that documents nicely, this was released by JPL just prior to the Curiosity landing, that documents this process that at that time was a completely new way of landing a rover on Mars. So that begins our surface mission. We are designing the system for 20 kilometers of traverse capability. So getting right into that, our first objective is to just study the geology of this landing site. So like any geologist, and these rovers are really just robotic geologists, we find ourselves in a new, unfamiliar environment. We open our eyes and take a look around, and we mostly start by taking pictures. And we look at the colors and textures and structures around us in the rocks that we see around us and try to understand what were the processes that formed these rocks. And so what environment do they represent? And for our landing sites, which I'll show you on the next slide, we're looking at extremely ancient environments, more than 3 billion years old in every case. Because this is a time when, as the previous speakers have said, we think Mars was much more Earth-like. There was a thicker atmosphere. It was much warmer and wetter than it is today. Mars today is a very inhospitable place. It's not impossible that it's currently inhabited, but it's much more likely that if there was ever life on Mars, we believe in any real diversity and biomass, that it was very long ago. And so we're going back to environments that are 3 to 4 billion years old and exploring those rocks to try to understand what were these environments. As we understand, was this a lake or a hydrothermal environment or some deep subsurface water alteration environment, we'll start to move into the next objective of the mission, which is the in situ astrobiology phase, where we ask the question, could this environment truly have supported life? We will select the areas within our landing site that we think give us the best chance of sampling rocks that have a high probability of having preserved signs of any life that was present, and also signs of environmental change. We're interested to understand Mars as a planetary system and how it evolved over billions of years. When we find those places, we'll sample them, and we'll drill, just like MSL, we'll drill into the rock several centimeters, but different from MSL, that takes powder in its drill and brings it into the rover to analyze with its instruments. We'll make a core, and we'll seal that core in a titanium tube and place that on the surface for a future mission to come with a rover that will bring a rocket, pick up our samples, put them into the rocket, launch them into orbit. The samples will now be in orbit, at which point a third mission can rendezvous with those samples and fly them back to Earth so that we can analyze them in Steely's lab or other labs here back on Earth and understand whether there are signs of life in those samples. And we think that's really critical to effectively really address this question of, was Mars ever inhabited? It's also a very important step towards the next objective, which is to prepare for future human exploration. In some sense, everything we do at Mars better prepares us to bring humans to Mars and bring them back safely. There are a few things very specifically that we maybe can get into later that we're doing to prepare for that human exploration. Thanks a lot.
So I have a question that maybe both of you can answer. There's a concern about contamination, right? How do we make sure that whatever samples we get back from Mars or potentially elsewhere are, if we do find little bugs in them, that they actually came from Mars and not from us?
Yeah, so contamination is a big one, and it's probably our biggest area of concern. It has been our biggest area of concern on the mission development so far. It's been the thing that we've spent the most time thinking about, the most time developing, and we have a set of requirements. So the entire mission lives in this requirement space. It's a very legalistic, sort of hierarchical system of the mission shall do this and shall do that. And one of our level 1 requirements is that every sample that we collect has to be free of any terrestrial organism whatsoever. So they have to be completely sterile. Now, that may seem easy, going to the doctor and using the word sterile, but building a robot to fly to another planet, drill a rock sample, and seal it in a tube without having a single terrestrial organism with very high probability of success in that endeavor is actually incredibly difficult and has never been done before.
How can we understand when we see the signature of panspermia? These extreme environments that permeate through the solar system, perhaps. You know what I mean. Yeah.
Yeah, so this is something we spent some time talking about today. We wrote this list of big questions on the board, and one of them that I wrote was panspermia versus second genesis, or however I wrote it. But just think about the scenario. If we're super successful with Mars sample return and we bring back a set of rocks and we do a bunch of analyses and we find multiple converging lines of evidence all confirming for us that there is evidence that Mars was once inhabited. We have fossil bacteria-like cells that have isotopic compositions and so forth. A whole set, good body of evidence. That will tell us that that there was at one time life beyond Earth, but it won't tell us what is, to me, the most profound question: has life ever emerged independently from life on Earth? And I think almost certainly so. I wouldn't be doing what I do if I didn't believe that life was very common in the universe. But there's a difference between believing that to probably be true and having clear scientific data that confirms that for us. And so to distinguish an independent origin of life from panspermia, that is, and I sort of skipped over the other part of the scenario, which is that the way you could get this set of observations in Mars samples without involving a second genesis of life on Mars is that if life emerged first and only on Earth in the solar system, and you had a giant impact on Earth, and you sent a piece of Earth rock and it landed into a habitable environment on Mars and inoculated Mars effectively, that is only a single origin of life is required for that within the solar system to have multiple planets inhabited. You could also run the thing in reverse, by the way. There's people who think we're all Martians. But solving that problem may best be in the hands of these folks, the exoplanet folks, because it's much harder to send life from one star system to another. So.
I think that when we finally have definitive evidence for life elsewhere, it's going to be a huge discovery. And I think it's going to really sort of change the way we see our place in the universe. When it becomes this sort of day-to-day fact, that's going to be really profound, because that'll sort of fundamentally weave its way into the fabric of how we understand our place in the universe and how society understands how we're placed in the universe. So I'm excited. I hope other people are too. I want to tell you the first part of the story of how we might detect life on other Earth-like planets orbiting other kinds of stars. Our solar system gives us an incredible diversity of nearby worlds that we can use to understand planetary processes and the different kinds of planets that might exist elsewhere. But even though there's a great diversity of potentially habitable environments beyond Earth in our own solar system, there's not another one of these. We don't have another Earth analog environment in our own solar system. And so the sort of holy grail for us as astrobiologists and exoplanet scientists is we want to find another Earth. We want to find another planet that is both habitable and globally inhabited in such a way that we could detect it across interstellar distances. But the great challenge that we face when trying to do this is that our images of exoplanets are not going to look like the beautiful video that I just showed you. Our first images of exoplanets are going to look something like this, right? We're going to get little dots, little points of light on our detectors. And so the great difficulty that we're facing is trying to extract information from these dots. Now here's a dot. This is an image of Earth itself that was taken by the Voyager spacecraft as it was leaving our solar system, and it turned its camera back when it was about 3.7 billion miles away from Earth. And it took this iconic image that Carl Sagan called the pale blue dot, where Earth here is suspended in this sunbeam. These images of exoplanets are going to look similar to this. Now they might not be pale blue dots, they might be other colored dots, but they're still going to be dots. And from these dots, we're going to be asking these profound questions, like, are these planets Earth-like? Are they truly habitable? And do they have life? So these are really, really big questions to ask a pale blue dot. How do you actually address this? What do you do about this when all you have to work with is a single pixel on your detector? Well, fortunately, there's actually another dimension of information that comes along with these dots, and that's their spectra. So if you split up the light from these dots into their component colors of the rainbow, including the colors that our eyes can't see, there's a whole new dimension of information that you can get. And these spectra are incredibly valuable and information-rich. Now, this is a cartoon that shows the spectrum of the sun, so the sunlight after it goes through Earth's atmosphere. So what's really cool about every atom and molecule in our universe is they let us know where they are in the universe because they have this very particular absorption spectra. So if you were to take a prism outside and split apart sunlight into a rainbow and look really, really carefully at it, you would actually see dark lines interspersed in the bright colors. And those dark lines are absorption features that are produced from absorptions by different compounds that are in either the atmosphere of our sun or in the atmosphere of Earth as the sunlight passed through Earth. And so by measuring the sort of fingerprints or barcodes of these different compounds, we can learn about what different things in the universe are made out of. What might you look for in the spectrum of Earth to understand, is our planet habitable? Well, one of the first things you'd probably look for is water. Water is really central to our definition of planetary habitability, and likewise, it's one of the first things we're going to look for when we look for habitable exoplanets. And Earth's spectrum is really awash in features from water vapor, and so this is really, really one of the key things that we're going after. We could also look for things like greenhouse gases that are like a blanket in our atmosphere, keeping our planet warm, similar to how this blanket is keeping this kitty cat warm. And I'm a cat person, if you can't tell. So greenhouse gases, if we could quantify their abundance from the planet's spectrum, we could then feed those abundances into computer models to try to predict what the likely surface temperatures of these planets might be. And we might even be able to see signs of geological activity. Geological activity is really important for maintaining planetary habitability over long timescales. So you could look for volcanically produced gases. You could look for things like volcanic ash. And you might look for time variability in these things that might indicate active volcanism. All of these processes are really important to understanding planetary habitability, and they're all things that we might be able to go after with future observations of exoplanets. But of course, detecting habitability is a little bit different from detecting inhabitants. How do you actually go from saying, okay, here's an interesting target that might have life, to saying, okay, here's a really interesting target that we think does have life? Well, in some ways, life kind of makes it easy on us because life is a planetary process that can modify its environment on a global level, the same way that geological processes can modify its environment, Earth's environment on a global level. And actually, right now in this room, we're all taking advantage of that fact because we're breathing oxygen. And the oxygen that we're breathing right now is a biosignature. It's a product of life. In particular, it's a product of oxygenic photosynthesis. Now, prior to about 2.5 billion years ago, our atmosphere lacked significant quantities of oxygen. So the oxygen that we're breathing and that's detectable in our spectrum has been a way for these oxygenic photosynthesizers to say, hello, we're here, to the rest of the universe for about the past 2.5 billion years. We could also look for gases like methane, which can also be produced by life, like microbes called methanogens that live in places like the guts of cows, although methane can also be produced by geological processes. When we think about biosignatures, it's always really crucial to understand biosignatures in the context of the global planetary environment to try to understand, are they more likely to be produced by non-biological processes in a given context, or more likely to be produced by biological processes in a given context? Thanks.
I think the one thing that's been most exciting to me has been what the Kepler Space Telescope has found and just the plethora, the huge number of planets it's discovered. When Kepler launched, we didn't know how many planets were out there, and we certainly didn't know how many planets that could harbor life are out there. And it's literally changed the way I look at the night sky. If I go out, I can count stars, and now when I count stars, I'm counting planets because we know there's at least a planet for every star. That's different. That wasn't true 10 years ago, and it is true today because of the Kepler Space Telescope. So this is one of the questions we often get when we talk about looking for alien life, is you're talking about alien planets with alien environments and aliens on them, so you should be thinking about alien signatures from those worlds. And that's what I'll be talking about, is how do we do that, especially in the exoplanet context, where the environments might be dramatically different from what we have on modern-day Earth. One is I call it like paint a planet model, okay, or paint a planet method. So here's the pale blue dot we've been talking about. But what if the dot wasn't blue because the life on the planet wasn't green? What if the life was a different color? Moving from left to right, you could imagine a plant life on a planet around a star where the star was much dimmer and didn't give off as much visible light energy as we get from the sun here at the surface of Earth. The plants on that planet might respond by having a variety of pigments, so they could get every single last ounce of or quantum of energy out of the photons. Ounce is not an energy unit. So every little bit of energy they could get out of the photons from that host star. So they might not be green. A verdant planet around that star might be black. You could also imagine planets that are more lush than Earth because of different nutrient availability or different continental and ocean arrangements. They might be totally covered in green and not really blue because there's more life covering the surface. And then the last one on the right is kind of the opposite of the one on the left. If you've got too much energy from the host star, the plants might be shedding more high energy blue photons away to prevent from overheating. And so they might reflect more blue light and end up blue. Now, the other thing that we've been doing to do this is to look back in Earth history. We're fond of saying that the most alien planet for which we have data is Archean Earth. Okay, this is a period in Earth history. If you went in a DeLorean or a time turner or a TARDIS or a hot tub and went back like 2.5 or 3 billion years, you better bring a gas mask. Not because there wasn't an atmosphere. The atmosphere was in place, but there was no oxygen at the time. Yet we have lots of evidence that there was life on the planet at the time, right? So if we're looking for life, and we know that for at least a third, maybe up to like 3/4 of the history of life on our planet, there wasn't enough oxygen in the atmosphere for us to detect with a telescope. That's a bad sign for us to, our attempts to look for Earth-like life on planets around other stars. So there's work actually led by Dr. Jada Arney on the types of signatures we might recognize on this anoxic, oxygen-free type biosphere. So that's one way we've thought about a different kind of biosignature that we understand only because we've looked back in Earth history and have thought about how life has evolved with co-evolved with our home planet. And by the way, we looked up the color of this planet on like one of those wall painting sites, like from a Home Depot or Lowe's type paint place. This is a pale apricot dot. That's the color that they assigned that particular planet. So that you can imagine how we might change the host star or change the chemical context of a planet and see how that affects what life would do. We want to generalize this so we could basically assign a model an environmental context and predict how biology would respond and figure out the best, most advantageous energetic reaction that biology would do and predict what it would produce as a byproduct we would see. Now, this is something we're going to be developing over the next decades with a huge set of collaborations. And as we're doing that, there's going to be folks like Olivier that are going to be developing the technology. So it's a difficult problem, but as they develop ground-based and space-based observatories to collect the data, we're going to be developing the frameworks to interpret it. Thank you.
So you guys both alluded to the fact that Earth did not always look Earth-like from afar, right? There was a billion or so years where it was a really different planet and would have looked different to an alien using a telescope and maybe trying to detect biosignatures here. How do we look for something that we don't know what it looks like?
Well, what we've started to do is look to, as Sean was alluding to, we've been looking to early Earth, because early Earth is an example of something that is by definition Earth-like, and we know it was habitable and inhabited, but it expressed in many cases these very different environments compared to the planet that we live on today. And so I think it's really important for us to look at all of Earth's history, not just modern day Earth, when we're thinking about detecting not just life, but also habitable environments elsewhere. Like a pale orange dot looks really different from a pale blue dot. It looks fundamentally different. And so if we're just looking for pale blue dots, we might miss this sort of spectrum of other kinds of worlds that are out there. So yeah, I mean, I would answer the question by just start with Earth's history.
We're designing a lot of our missions around that oxygen-methane pair. But the generic way to be as inclusive as many biospheres and biosignatures as possible is to have as broad a wavelength range as possible. So when Jada was talking about that spectroscopy, we want to go as blue as we possibly can, like bluer than the bluest light we can see out into the ultraviolet, and as red as we can possibly see out into the infrared, past redder than our eyes can see. If we had if it was possible, we'd go all the colors, right? But there's always a discussion about what's actually implementable. So it's like we've got these discussions going on with engineers, and I'm sure the Mars scientists and the Icy World scientists are doing this as well. You have to eventually fit in a rocket, both that can lift the mass and the mass you have on the telescope up into space. And you can't give too many technological challenges to one generation of missions either. So there's always like a balance there, but we're always pushing for as broad a wavelength coverage as possible.
Hi. I had a question on the note of purple stars, pale purple dots, and the purple stuck in my brain, and pale green dots. So the reason I mentioned stars is, as many of us know, purple stars and green stars do exist in reality, but we don't perceive them that way. The sun is a green star, but we perceive it as yellow, and purple stars are perceived as blue. So when I think of a pale green dot, I think of Proxima b, and it's not because of leafy greens or a high pollen count, it's because of aurora. So if you look at Proxima b with naked eyes, will we see it as green, or will we see it as something else?
I think we need to do that experiment to find out. I mean, there has been a paper out of the University of Washington led by Rodrigo Luger and Jake Lustig-Geiger, who are looking at the potential for auroras to occur on Proxima Centauri b. I want to do the experiment. I want to find out.
Well, that's one of the fun things about this moment is like you can turn any of our ideas of how processes might operate in these different contexts and change that from something we would just like write a model or a paper about. But now it's like a testable hypothesis because we can make a prediction that we can test with observation, right? And that's basically what you just did with that question, which is like, for me, that's the cool moment of this generation.
Well, the day the discovery of life beyond Earth hits the front of a newspaper is going to be a big day. It's going to be a very big change in the way we actually view ourselves, similar to the way that we discovered, for example, that Earth was not the center of the universe. I work on designing instruments for telescopes so that we can meet this challenge. We can image planets around other stars. And we are trying to do this for both ground-based and space-based telescopes. So, of course, we like to use large telescopes. So here I have a picture of one of the telescopes I work with. It's the Subaru telescope. It's in Hawaii. So the real goal, as was just explained, is we want to do direct imaging. So we want to take an image of the planetary system where we can see the star, and a little bit away from it we can see the planet. So we can take the light of the planet free of most of the light of the star, and we can take a spectra of that light. So we can decompose it in colors, in wavelengths, and by removing most of the starlight we can get very good signal. And the goal is to get a spectra like the one that's shown on the right. That's a spectra of Earth, which was actually taken by looking at the moon. If you look at the thin crescent of the moon, the part that's not illuminated by the sun is actually dimly illuminated by Earth. So you can take a spectra of that light, you can divide it by the part that's illuminated by the sun, and you get the reflectance spectra of Earth, as shown on the right. When you do that, you see a very rich spectra. You can see water, oxygen. You can see that there's also ozone in the blue UV. You can even see that the sky is blue. There is scattering from our atmosphere, so there's excess of light on the blue side, which is on the left side of this spectra. Here's an image, actually, of a habitable planet. You may not be able to see it from the back. So I'm showing you here an image of Saturn, which is not a habitable planet, taken by the Cassini spacecraft, which is now dead. But it was, at that time, it was flying in the shadow of Saturn, looking back towards the sun. And there is a small dot, which is labeled, which is on the bottom right of the disk of Saturn, where all of us are on that pixel. If I zoom in, that's us. The point that this picture is making is that Earth, as soon as you leave our solar system, is pretty faint compared to the sun. It's pretty small. It's hard to see. The contrast ratio is about 1 billion or 10 billion to 1. So basically the sun is about 10 billion times brighter than Earth. To make things worse, if we try to do this on if we look for other Earths, the other Earths are going to be very close to the star. So it's a significant technological challenge, and a lot of us are working very hard to meet this challenge. So on the ground, our community is very actively developing on current telescopes the technologies that will allow us to image habitable planets. So here is one example. That's a project I work on at the Subaru telescope. So the telescope is shown here in this picture, and in the foreground you can see the picture of the instrument that we're still continuously building to test these new technologies. This is another example, the Magellan telescope, which Carnegie is involved in. And there's a similar project, MAGEO-X, which is developing also the technologies to solve this challenging problem. And what our community is doing is basically using the current large telescopes to sort of sharpen our tools so that when larger telescopes come online, we will be ready to image habitable planets. To summarize, we're actually gearing up to target habitable planets, and it's going to take a combination of three things: the technology development that we're doing, the large telescope, and also we're going to be smart about the targets we're going to be looking at. We're going to look at habitable planets from the ground with around stars that are fainter than the sun, because those have a more moderate contrast and make a better use of the large aperture of the telescope, which can resolve small angles. And in parallel, in space, NASA is another space agency are thinking hard, studying concepts for large space-based telescopes that will target planets around sun-like stars. So thank you. This is a picture, by the way, of a video of the LUVOIR telescope, which is the largest concept that NASA is studying. So it's a sort of scaled-up version of James Webb, but optimized for high-contrast imaging. Thank you.
What's the timeline for potentially getting a direct image?
Our community is basically trying to be ready for the time when the large telescopes are online, which is roughly 10 years from now. So we're really aiming at that time scale.
Hi, I was wondering, how do we distinguish planets that, like us, start out inhabitable, but might in the future be habitable? Like us. So based on how our planet developed to be habitable, basically.
Brilliant question.
Really good question.
Well, I think the starting point is understanding what the star does to the planet. And one thing we understand is the stars tend to get basically fainter as the planet as the age goes on. So we know the habitable zone is not a static place. It's moving. It's moving as time goes on. And then there is all sorts of questions that are. I think I'm trying to answer your question by saying we don't really know, in ways that don't sound that bad. But there's a lot of things happen with the evolution of the planet itself. One thing we're worried about is maybe going the other direction than your question, is that maybe the atmosphere will escape and the planet that used to be habitable no longer stays habitable. That's in part what happened on Mars. And there's many scenarios. Things can go either way. One way that planets used to not be habitable and become habitable is the solar system becoming a more quiet place. Early on, you get bombarded by big rocks, and that's obviously not nice. So you need to sort of wait until things are a little bit quieter so that you have a more stable environment in which life can evolve.
I think when we begin to be able to think about ourselves in comparison to something else out there, it has the effect of trivializing the differences among us and making us see ourselves first and foremost as Earthlings. And that's a good thing, because we have lots and lots of problems and challenges on this planet that don't respect national boundaries. And so the more we see ourselves as the same, I think that sets us up to be better able to solve these challenges in our future.
Humans have been on a journey to understand where we came from, who we are, why we are, how we fit in, and of course, is anybody else out there for millennia. And some of us are interested in finding mathematicians as well as microbes. And so over the course of my long career, there have been two extraordinary game changers, which you've heard about earlier tonight. The extremophiles, showing us that life can live in an enormous range of environments, and exoplanets and potentially exomoons, showing us that there is a lot of real estate out there. So we are at a point where we can actually begin to understand whether any of that potentially habitable real estate is inhabited. And we've heard a discussion of how biosignatures might allow us to answer that question. But you also might consider technosignatures, right? Detection of someone else's distant technology, not just the biology of life. So our technology is observable over interstellar distances, and theirs might be as well, even if we don't know what that technology was developed for, even if it's doing something very, very bizarre. We still, with a concerted effort of searching through optical, infrared, radio, parts of the spectrum, we might in fact detect that technology. And so we began this process over 50 years ago, and although this is an image of the telescopes at the Very Large Array, that first search was done in Green Bank, West Virginia by Frank Drake in 1960, using a telescope that was designed by Judge Tatel's father, the Tatel telescope at Green Bank. So it has a long history with Carnegie. And at the beginning of the century, we opened up our searches for signals into the optical. And now that the technology is becoming available, we're beginning to explore the near infrared. And what we've been looking for are artifacts, engineered signals, signals that, as far as we know, nature can't produce. So if you start to think about all the places where you might find signals and the characteristic of those signals, and you say, that's the volume I want to search. Okay, I'm going to give you an analogy of what we've been managed to do in 50 years. So set the volume, and it's actually about 9-dimensional, set that equal to the volume of the Earth's oceans. And how much have we searched? Well, only about 1 12-ounce glass. Right? Because it's a big universe out there, and there are a lot of places to look. But we are really excited because our tools for looking are getting better, and they're getting better exponentially. And what we've realized is that we have been totally blind to a class of signals that are transient. We know that in order to find signals that don't persist for a long time, signals that might come once and then not repeat for an undetermined amount of time, we have to be looking at all the sky, all the time, and preferably at all wavelengths. Now that gets to be as challenging as the spacecraft and the telescopes that you're trying to build. Big job. But we're beginning right now, for the first time, to be able to tackle that problem, at least in the optical. And the other thing that's going to help us enormously in the near future is machine learning. Imagine the distant, somewhat more distant future when we're beginning to get those images of pale blue dots. And we've heard that it's really hard to tell what that dot is giving us. But if you begin to think about technology and how technology might change the direct images of those pale blue dots in ways that are detectable, you can begin to really scratch your head about these technosignatures. Well, you know, they might be doing something that would give you strange reflections and glints. They might be organizing and changing their planet in a way that makes it at the wrong temperature for where it is with respect to the star. They might have decided that it's really not a lot of fun to have too many hurricanes and extreme weather, and they can maybe engineer that out of their planet. I'm not sure how you tell that at a distance from a pale blue dot with the kinds of timescales involved. But how about they decide that they really like a particular temperature, and they want the whole planet to be that temperature, and they might manage to remove the temperature gradient from equator to pole. Maybe we can sense that, even though it's just one image. When you begin to talk about technosignatures, there is another aspect that normally doesn't get discussed with respect to biosignatures. That is, if SETI, or search for technosignatures, is going to succeed, there have to be two technological civilizations close enough to be detectable. But that's not just close enough in space, it's also close enough in time. You have to have two technological civilizations
of some longevity lined up in this deep 10 billion year history of our own galaxy, and they have to overlap. And that's never going to happen statistically unless technologies, on average, last for a long time. That is, that technology turns out to be a stabilizing influence, and so they can be long-lived. We are a very young technology in a very old galaxy, and we don't yet know whether it's possible to become an old technology. It's a big question for us. For me, that's why the search for life beyond Earth, biosignatures, technosignatures, SETI, is so very important, because it gives us an opportunity to hold up a mirror to all of us here on this planet and make us stretch and think about how we, relative to them, we are all the same. It trivializes the differences among humans. It makes us expand our perspective to see ourselves as all the same as Earthlings. And I'll leave you with the last words from Caleb Scharf, an astrobiologist at Columbia University. And Caleb reminds us that on a finite world, a cosmic perspective is not a luxury, it's a necessity. Thank you.
Jill, you talked about this, the amount of real estate out there for us to search is as vast as Earth's oceans, and we've only, with SETI, we've only looked at a tiny glass of it. And you've been a driving force in that for a long time. Do you feel more optimistic about the possibility that one day we're going to scoop up something interesting in that glass than you used to, or less optimistic?
Well, I have to say that the universe today versus the universe that I began to study as a student, the universe today appears more bio-friendly than it used to. We don't know that it is. That's our challenge to decide. But we didn't know about any planets beyond our solar system when I was a student. Could have turned out that there weren't hardly any of them out there. But that's not the case. More planets than stars. So, yeah, I mean, the arc is positive as far as I'm concerned.
Dr. Tarter, as opposed to your other colleagues who are just searching for life, you're searching for life like us. But I know what can happen on an evolutionary basis in 13.7 billion years, and I know Fermi's paradox. How do you rationalize what you're trying to do, searching for beings like us, with the perhaps improbability of beings like us searching for us over the time available in the universe?
Well, first of all, probably we should define Fermi's paradox. Fermi came to lunch at Los Alamos and said, "Where is everybody?" And his implication there was, if there had ever been another technological civilization anywhere or any when in the galaxy, then clearly they would develop over a very short time the capability for interstellar travel. And then no matter what your model was of how colonization might take place, they would have spread throughout the galaxy in a time that's short compared to the history of the galaxy. But he said, they're not here. So that only explanation is there can never have been another technological civilization. We're the first. And I think the problem with that whole analogy is that we can't say they're not here. We have so poorly explored even our own solar system. There could be all kinds of small artificial intelligent nanotechnology around that we haven't detected. I mean, it's hard to emphasize how big space is, how little of it we've looked at. So my answer is, how should we know? We've hardly begun to look.
So let me ask you to dream a little bit.
We haven't been dreaming yet?
Yes. Let me ask you to dream a little bit more. We find a signal of life. It's credible. So SETI finds this tomorrow. What do the rest of you do? How do you change your view going forward? Technologically, search, etc.
I think we'd look harder. You know? I mean,
we got to catch up with SETI.
You count 1, 2, infinity. So as soon as you find number 2, you go looking for all the others that you know have to be there.
Yeah. If we find one other one, we know that life can't be rare, especially if it's around a nearby star.
I'm curious also, I mean, we've talked about the potential philosophical implications of a second genesis of life and the scientific implications. Are there kind of like practical global implications, you think? Like, what does this mean for society?
Mm-hmm.
I think it has to unite us as, I mean, Jill probably said it best on the panel, as Earthlings. You know, I think the people up here and people who study astrobiology, this is the view that we were probably just born with, that we see ourselves all on this planet as you know, one big species and one big you know, inhabited planet, and we're all Earthlings. But we, as a species in society, we don't tend to behave like that, especially these days, and there's so much division, and so I think astrobiology generally is such a naturally exciting topic for young people. So it's great for it's great to pull young people into science. It's great for that. As we've discussed, the kinds of science that we want to do, the kinds of goals that we have, are these multi-generational cathedrals of science, and that require such huge resource commitments that we may be obligated to pursue them multinationally. And so for many reasons, I think the finding of evidence of life beyond Earth would have implications. But clearly, we don't even have to find it to realize some of those benefits. It's just the search, the doing of it, has major implications for society.
And actually, on that note, I think we're almost out of time. So I want to invite Dr. Andrew Steele from the Carnegie Institution to give some closing remarks.
First of all, I'd like to thank Sarah as your moderator and the panelists this evening. Please give them all your appreciation. I hope that you get a sense of not only how we're looking for this life, but how the technology is developing, that you get a sense of the debate between the scientists as to what are the most important things to look for and how we're going to look for them, and the sense of cooperation and collaboration between scientists that is necessary to answer this question. Are we alone? And I'm going to borrow a phrase from Jill, where she's saying we're all Earthlings. Now, I was struck by the parallels of that to a talk I heard by the Dalai Lama, where the first thing that the Dalai Lama said is, number one, we are all human beings. Everything else is details. And I want to thank you all for your wonderful questions and your participation this evening. Thank you very much.